Variable number tandem repeats. Variable number tandem repeats are short sequences which are organized in the form of tandem repeats on the DNA. These type of the repeated tandem DNA sequences are present on almost all the chromosomes. They show variation in length between the individuals. The size of the VNTR varies between 7-8 nucleotide up to few tens of the nucleotides. Each variant in case of VNTR acts as an allele. So there is difference between the size of the alleles. So due to that will be due to the size of the variants or the nucleotides which are present in the VNTRs. If we see here how to understand the concept of VNTR, these are the blocks. Each block that represent to the repeated DNA sequence. We can say that in this individual, that is individual 1, these are block, they represent to a sequence of DNA, and same sequence of DNA will be repeated third time, fourth time, fifth time and sixth time. So you will say that in individual 1 there are 6 repeated regions of VNTR. So they are clustered together and they are oriented in the same direction. In case of individual 2, the same DNA sequence is of 4 times. In case of individual 3, the same DNA sequence 3 times. In case of individual 4, the same DNA sequence is of 5 times. So we can say that these blocks, they represent to the repeated DNA sequence, and these repeated DNA sequence which are shown in the form of blocks, they vary between the individuals and they are usually clustered together and they have same orientation on the DNA strand. So how to study the VNTRs in the individuals or in different organisms? Usually the methodology of the VNTR is that restriction enzymes are used to cut the VNTR blocks from its specific site and analyzed by RFLP. So these blocks, they are cut with the help of restriction enzymes and they are analyzed with the help of restriction fragment length polymorphism. So blocks which are called as VNTR, they are amplified by a technique which is called as polymerase chain reaction. The amplified products are run on the gel electrophoresis, which also determine the length of the VNTRs. So if we summarize the methodology, the restriction enzymes are used to cut the VNTR blocks or VNTR regions with the help of certain restriction enzymes. These blocks, they are amplified with the help of polymerase chain reaction. They are run on the gel and to determine the length of these blocks or VNTRs and variation between different individuals. So this is the diagram where we can see the variation in patterns of bending. Here we can see that these are the bending pattern of different individuals, and we can see that the bending pattern is different in each of the lane. If we take the example of this individual, this one, here we can see one band is here, second band is here, and the third band is here. While in the next lane we can see one band is here, second band is here, which was not present in this individual. So here is no band. So second band is here, there is another band in this row, and we cannot see that band in this individual. So with the help of these bending pattern, we can identify the variations between the VNTRs. So what is the importance of the VNTRs? VNTRs actually used for the linkage analysis studies, particularly for mapping the genomes or for mapping different genetic regions. Importance of VNTR: if we remove the extra DNA from outside of the blocks and digest that particular DNA where VNTR are present and amplified, so with the help of those VNTRs we can study the bending patterns between the individuals or the DNA of the plants or crops. Some basic principles which are related to the VNTRs. In case of VNTR, there should be identity matching. Identity matching means if the DNA of a single individual is amplified twice or it is studied 2 times, so the bending pattern each time of that individual should be same. This is called as identity matching. While on the other hand, inheritance matching. Inheritance matching is if a person is studied, he has a certain bending pattern. His bending pattern should be consistent with the bending pattern of his mother and father, because the DNA is inherited from both from the father and the mother. So the bending pattern of that particular individual should be according to the bending pattern of the parents, that is the father and the mother. Applications of VNTRs. There are different applications of the VNTR, like in microbiology, DNA fingerprinting, DNA and genetic diversity, in forensics, and mapping certain diseases.